Okay, so yesterday we spoke about the structure of the nephron. If you remember, yesterday we spoke about our kidney is made up of millions of nephrons, right? And each nephron has two parts: the renal corpuscle, okay, which is the head-like part. This one, let me get the pen. This part is the renal corpuscle, okay, and then we have a tubule like part. So this is the tubule like part, right, which is called the renal tubule, right? So then we said that this head like part called as the renal corpuscle is further made up of glomerulus, these capillaries called glomerulus, and then it's surrounded by the Bowman's capsule, okay, which contains the filtrate. And then filtrate moves into the tubule called as a renal tubule, which is made up of three parts: PCT, proximal convoluted tubule, then we have the lobe of Henle and the distal convoluted tubule. And then we have the collecting duct, of course, at the end. Okay, that's what we learned yesterday. But in today's class, we'll talk about the different types of the nephrons. We have nephrons like cortical nephrons and the juxtamedullary nephrons. So what are cortical nephrons? Cortical nephrons are, as per your book, which are located entirely in the cortex. You know what is a cortex area, right? If this is the kidney, right? So cortex is this outer area is called as cortex, the superficial area. This area will be called as a cortex, right? So as per your book, it says that if you, this is a diagram from your textbook, it says that look at this. This cortical nephron is totally present in the cortex, but that is actually not that's partially true. That's not actually correct. They're not totally into the cortical nephron, and they're not totally into the cortex area. Their loop of Henle, this loop of Henle goes into the medulla, but not deep in the medulla. This is the cortical area, this is the medullary area, right? So their loop of Henle is present in the medulla, but not very deep in the medulla. Let me show you a picture for that. Look at this. This is a diagram which is not from your textbook. Okay, showing the cortical nephron. See, this is the Bowman's capsule, and this is then the proximal convoluted tubule. And look at this. Its loop of Henle is going down into the medulla, but not very deep into the medulla, right? And then the distal convoluted tubule is present again in the cortical area, and this is finally the collecting duct. Okay, so but in your exams, if you get a question, so you will write as per your textbook that yes, cortical nephron is completely present in the cortex. Is it okay to everyone? Did you all understand for the exams what you need to write? But you have to remember that yes, the loop of Henle goes into the medullary area, but not very deep. Okay, and almost 80%, 80 to 85% of the nephrons are the cortical nephrons in our kidney. Okay, and then what are the juxtamedullary nephrons? Juxta means next to, I mean next to or besides. Okay, so next to, these nephrons are present next to the medullary area called as the juxtamedullary nephrons. Okay, so they are present close to the medulla, next to the medullary area. Okay, and let me show you the nephron. How does it look like? Okay, this is present in the cortex medulla junction. Look at this. This is this is a juxtamedullary nephron. So this is the cortical area. This is the medullary area. So it lies in the at the junction of the cortex and the medulla, as you can see here. Okay, so this is their Bowman's capsule. This is their PCT, right? And look at this. Their loop of Henle goes deep down into the medulla. Okay, for the juxtaglomeralis medullary nephron, if you see here, look at the diagram. Okay, it's loop of Henle goes deep down into the medulla. This you have to remember for the juxtamedullary nephrons. So like 20% of the nephrons belong to this category. Okay, so nephrons which have long loop of Henle, okay, helps our kidney to excrete either a very dilute urine or a very concentrated urine. Okay. So did you all understand the difference between the cortical and the juxtamedullary nephrons? Most of the nephrons in our body belong to which category? Yeah, they belong to the cortical. Yes, cortical nephrons. Absolutely, Haifam and Hadil. Okay. Then we have the capillary beds. Okay. We have two types of capillary beds in each nephron. Okay. One is called as the glomerulus. Okay. Network of capillaries, blood capillaries. And then next one is the peritubular capillary bed. Okay. Glomerulus, if you remember yesterday, I showed you the structure of the glomerulus. This is the glomerulus. Look at this. This is the glomerulus. Look at this. Okay. If you remember yesterday, I told you that they arise, it, it, the efferent arteriole, okay, enters into the Bowman's capsule, right? And that efferent arteriole is basically fed by the cortical radiate artery. So it feeds the glomerulus. Okay. So the efferent arteriole feeds the glomerulus. And then if you remember yesterday, I told you it's a little larger in size as compared to this one, which is a little smaller in size. Right? The efferent one. This is the efferent coming out, efferent going in, right? And this has a significance. Why this is larger and why this is smaller? The reason is when blood enters through the efferent arteriole, it is basically, blood has very high blood pressure, okay? And then because this area is very, very small, the efferent arteriole is very small, this further increases the blood pressure, okay, because the efferent arteriole is very smaller in the diameter, okay, because there will be a lot of resistance to the outflow of the blood, okay. And this we'll discuss in a while. Why do we want here more blood pressure? Because this will help us in filtration, because this capillary is totally meant for the filtration, to filter the blood, okay. So the filtration will take place here in the renal corpuscle. So look at this. The afferent is bigger than the efferent one, larger in diameter than the efferent one. That is the same thing which I explained you yesterday. The fenestra, the capillaries have, smaller holes through which things can move out, okay, from the capillaries, except the RBCs, because they're big enough, they cannot move through these fenestras. Peritubular capillary bed. This is a capillary bed which you can find here when the efferent arteriole comes out of the glomerulus, okay. It gives rise to another capillary bed. Let me get another color. Look at this. It gives rise to another capillary bed which is continuously hugging the whole renal tubule, right? And this capillary bed is at very low pressure as compared to the glomerulus. Okay, they are also porous. Okay, and remember, they are not meant for the filtration. We'll talk about that. They are mostly meant for the absorption instead of filtration. Okay, and finally, they pour their secretions into these veins, right? So the cortical radiate vein, and then which forms the arcuate vein. Okay, so this capillary bed is called as a peritubular capillary bed. So we have two types of capillary bed present in the nephron. Okay, then comes the important part called as the urine formation and the characteristics. What is this? This is basically how urine is formed, the physiology of the urine formation in the nephron. Did you all understand? Is it clear to everyone? Okay. Okay, now In the urine formation, we learned that this is very important part of this chapter. Okay, in the urine formation, we have three important processes. Okay, Gadi, no problem. Okay, so here we have in the urine formation, we have basically three important processes. One is the glomerular filtration, another one is the tubular reabsorption, and the third one is tubular secretion. We are going to discuss about all three of them in detail now. What is the glomerular filtration first? So this is just the overview of these three processes. Then we will discuss about these in detail. Glomerular filtration is mostly, as if you remember, I told you that it takes place in this part, in the renal corpuscle part. So here the blood is simply being filtered in the glomerular filtration. And we already discussed that the capillaries here are very, very porous over here, right? So everything will move out except the RBCs and except the proteins into this Bowman's capsule. Then the fluid will move further towards the renal tubule, and where the what is the tubular reabsorption? Where the things will move from the tubule into the blood. Very important. The direction is very important. In the tubular reabsorption, we things move from the tubule into the blood. And I'll tell you in a while why do they move into the back into the blood when they are filtered here, why they move back into the blood. I'll explain you in a while coming, okay, in coming slides. And the last one is the tubular secretion. In the tubular secretion, this is very important to remember. Absorption is movement of the filtrate from the tubule into the blood, as you can see here, while as secretion is the opposite of it. Okay, secretion is when things move from the blood again into the tubule. Those things could be anything, any waste material, any drug which has not come out yet from the blood, that can still move out into the tubule, which is called as a tubular secretion, right? So these are the three main processes of the urine formation. Very important, all three of them. Let's start with the filtration. Filtration is simply, first of all, where does filtration take place? It takes place only in the Bowman's capsule. Point number one. Point number two, what is filtration? What happens in filtration? We all know that the main function of the kidney is to filter the blood, right? So in filtration, whatever is present in the blood here, excuse me, whether we have waste products, whether we have anything, like drugs or anything which we need to filter out of the body, right? So everything will move into the Bowman's capsule. So it's not only about the waste products which will move in the filtration, you will see that the entire plasma almost will move into the Bowman's capsule. Okay, and most of the solutes, almost not entire plasma, we have like most of the solutes present in the plasma. Okay, that can be glucose, that can be hormones, that can be anything which we need actually in the blood. All those solutes can move into the Bowman's capsule. Okay, so we define filtration as movement of water, or you can say plasma, okay, and the solutes present in the plasma, okay, except the RBCs and except the plasma proteins, okay, across the wall of this glomerular capillaries into the Bowman's capsule. So now the fluid is where? In this Bowman's capsule. Look at this. The fluid is in Bowman's capsule. Look at this. Now you might ask me, how does it move? What is the reason behind that? The reason behind is simple. It's a passive transport. It does not need energy. Okay, because we simply have the blood here, which is coming into this capillary network, is coming straight away from the heart. Okay, and it is at very high blood pressure. So blood pressure is very, very high. Okay, And plus, if you remember, I told you efferent arteriole is bigger in size, while as efferent is smaller in size. So efferent arteriole, the diameter of it is smaller. So as a result, there will be a lot of resistance when the blood will try to come out. Okay, so that's why there's very high blood pressure within the glomerulus. Okay, and it's simply from high pressure area to the low pressure area, things will move out. And this is called as the filtration. Are we all okay about filtration? Any question about filtration? So what is important here? Blood pressure plays a very, very important role. God forbid if a person has low blood pressure or has loose motion, has less fluid in the blood, so the kidney filtration will not take place in the Bowman's capsule. And and the result, we will produce less urine or no urine, which is very dangerous for the body. That is why maintaining blood pressure is very, very important. Okay. Now, second is tubular reabsorption, right? Now you might think that why do we need to reabsorb the thing? What is tubular reabsorption? Reabsorption is simply movement of the substances. As you can see here, the filtrate is now here. From the Bowman's capsule, the filtrate will move now towards the renal tubule, right? Excuse me. Now, what is the function of reabsorption? Reabsorption is that when we move the filtrate here, okay, into the Bowman's capsule, excuse me, excuse me. When we move the filtrate here in the Bowman's capsule, this filtrate, as I told you, it might have everything. It might have a glucose, fructose, amino acids, vitamins, hormones, any any important ion like sodium, potassium, calcium. But do we need these hormones or fructose or glucose to go out of the body? Yes, ladies. Should they be sent out of the body? The glucose, hormones, vitamins? Of course not, right? Of course not. We want them back, isn't it? So tubular reabsorption is the process, or you can say movement of important substances like your glucose, fructose, amino acids, hormones, which are there in the filtrate into what? Okay, into the renal, from the renal tubule into the peritubular capillaries, because we have capillaries present here. Let me get a pen. We have capillaries present here. We have capillaries present here. We have capillaries. We just spoke about the peritubular capillaries, right? I showed you the diagram of the peritubular capillaries. Okay, look at this. Look at this. So things which we need back, okay, they will go from the tubule into the blood. This is called as the reabsorption. So you can say reabsorption allows the movement of very useful substances to return back to the blood as the filtrate is flowing through the renal tubule, right? It is said that almost 99% of the filtered water and many solutes come back to the blood in the tubular reabsorption. Things like glucose, amino acids, they're completely reabsorbed back into the blood. Okay, and imagine, God forbid, if a person loses glucose through the urine or loses hormones or vitamins, okay, through the urine, or so that is very dangerous condition. So they should be retained back into the blood. Now you might think that what is the process? How does it take place? Okay, what is the is it active transport or a passive transport? You have to remember that it is everything. It's like a simple passive transport, like a facilitated diffusion, or we need to spend energy. So it's an active transport, and water will flow back into the blood by the process called as osmosis. Yes, and diabetes mellitus, right? So all the solutes are reabsorbed into the blood right? And water will automatically follow by the process of osmosis, right? So what are the things which cannot be reabsorbed? Okay, the things which are not reabsorbed are wastes. They will still stay in the filtrate over here. They are still part of the filtrate. They will not go into the blood. And what is the use of filtration? So wastes are still there. Uric acid, drugs, right? Excess ions, they are still over here. And the second question is, where does this reabsorption takes place? It takes place throughout the tubule, from the PCT here, okay? It takes place in loop of Henle, it takes place in DCT, everywhere, okay? So it takes place in all the parts of the renal tubule, called as the reabsorption. Then comes the third step, called as the secretion. Secretion is like the opposite of it. Let me show you here. It's the opposite of it. If you can observe in this diagram, give me a minute. See, reabsorption is from the tubule into the blood. So secretion is whatever is there in the blood, it will move in the opposite direction to the reabsorption, right? So because there might be some substances which have not yet come out in the filtrate, maybe some drugs are still there, maybe some creatinine is there, right? So secretion is very, very important. So it gives the kidney these substance, a chance so that these substances can go out of the blood into the filtrate and become part of the filtrate. Okay, and secretion is very, very important because it's the secretion which will maintain the acid base balance of the blood. Just in case if a person has acidosis, right? So quickly your kidney will send more and more hydrogen ions. What? Where? If you have sorry, if you have then hydrogen ions will move out. It will retain more and more bicarbonate ions if the person has acidosis here. So bicarbonate ions will be secreted into the tubule. Or if a person has, for example, alkalosis, okay, very high pH, then what will happen? More and more hydrogen ions will be taken back or secreted into the kidney tubule. So that is why I said that secretion helps in maintaining the acid base balance of the blood. So what is secretion? Simply movement of things from the blood into the renal tubule. So what is the reabsorption? Reabsorption is the movement of fluid or important substances or important solutes, okay, from the filtrate back to the blood. So these are two opposite processes, secretion and the reabsorption. Okay. So did you all understand the three processes? Any question about it? Filtration, reabsorption, and secretion. So why secretion is important? Getting rid of the wastes which are not already in the filtrate, or removing excess ions or drugs, or maintaining acid-base balance, as I told you, right? So that was about the urine formation. Now we'll talk about what are the nitrogenous wastes present in the body. Okay, we have wastes like urea, right? So nitrogen is highly toxic, especially to the brain. The ammonia is highly toxic, especially to the brain. Okay, so they are not at all reabsorbed. Okay, so they remain in the filtrate and are thrown out in the form of either urea, okay, or uric acid or creatinine, because all of them are dangerous for the body. So we don't want them into the body, so they're filtered out. Okay. So in almost 24 hours, if you remember, I told you that almost 200 liters of blood is being filtered, but only 1 to 1.8 liters of urine is produced. Okay, and we need to know that there is a difference between the urine and the filtrate, right? Urine is basically what we don't need, right? So what, for example, when the filtrate has lost, okay, most of its water, nutrients and necessary ions through reabsorption, whatever is left out then, be it the urea, uric acid, waste, nitrogen wastes, that will be in the urine. But what is filtrate? Filtrate is which has everything that the blood plasma has, except the RBCs and the proteins. Because when things were filtered over here, okay, so everything was moved into the Bowman's capsule, whether it was a glucose, fructose, excess ions, calcium, vitamins, hormones, right? So it has all good things, filtrate. Okay. And then we have the solutes normally found in the urine. We can find sodium ions in the urine, potassium ions, the excess sodium ions or the potassium ions, urea, uric acid, creatinine, ammonia, highly toxic to the brain, bicarbonate ions, right? And the things which should never present in the urine, which shows that there is some problem in the kidney, because kidney will never filter out glucose, albumins or blood proteins, RBCs, hemoglobin, WBCs, bile, they should not go out of the body because we need them back in the body. So, in which segment of the nephron do the following processes will take place? Where does filtration take place, everyone? Yes, Haifa, Hadil. Where does filtration take place? Yes, absolutely. Renal corpuscle. Very good. Yes. Okay. What about reabsorption? Yes, Rava, very good. Hadil, in the tubule. But where in the tubule? Tubule has three parts: proximal, loop of Henle, and distal convoluted tubule. Where in the tubule? It has all three parts. Remember that. It takes place, yes, all three parts. Secretion. If you remember, I told you secretion will not take place where? In the loop of Henle. Okay, it will take place only in the proximal and distal and the collecting duct. And excretion means, that means urine has to go out now. Okay, now it takes place in the collecting duct. So this is the excretion. Okay, now that was about the physiology of the urine formation. And now we'll discuss about the ureters. Okay, so what are ureters? Does anyone know what are ureters? Ureters are something, if you see here, this is the, we have kidney over here. Okay, a kidney over here. So ureters are basically which connect the kidney with the bladder, right? These are the ureters. Look at this. Okay, so they're totally continuous with the renal pelvis. Okay, these ureters. Okay, and one thing which is very important, which you have to remember here is if you observe this diagram, they are entering the bladder from the posterior side. Look at this. These are two ureters, and this is one spot where they're entering. This is another spot where they're entering into the bladder. Okay. So, and then we have one more spot over here, this one. Okay. And what happens? So, this basically, it results in the formation of this trigonal area. Okay. The triangular area within the bladder. So this part is usually infected, gets infected. Okay. So most of the times when we say infection, so this trigone area is mostly infected, this part. Okay, let me show you the diagrams very clearly. Once again, let me erase this, as you can see here. Right? Look at this. This is one orifice, one hole, This is another hole, so where the ureters enter, and then we have another hole, this one over here. Right? So internal urethral sphincter is present over there. Right? So this is the trigone area, which is mostly infected because microorganisms can simply enter through this part and affect the area of the bladder. Okay, and then what? Then how does urine move from the ureter into the bladder? It's simply the what? The gravity. Gravity plays a very important role in in pulling the urine towards the bladder. And of course, in I told you in last chapter, the peristalsis not only takes place in the GI tract, the peristalsis also takes place in the urinary system as well as in also the reproductive system. Okay, this is the main thing by which things can move here. Okay, so that was about the ureters. Look at the ureters. Look at these are the ureters. Look at the ureters over here, right? And this is the there here we have the trigone area. As I told you, it has special importance because infections are mostly present in this area. Okay, then we have the bladder. Okay, so bladder is this is bladder. Okay, so can anyone tell me which tissue surrounds the bladder internally? Which epithelial tissue is present internally in the bladder? I think we did that in last semester. Epithelial tissue, which is very special epithelial tissue found in the bladder. Yes. Transitional, yes, Rava, absolutely. Transitional epithelium. If you remember last time, I told you that this is a very special kind of epithelial tissue which is present in the bladder, surrounding it internally. What is the specialty about this transitional epithelia? It can, what can allow exactly, it allows for the expansion. Absolutely, very good, you're right. Both of you are right, absolutely. Okay, because your bladder, once it's full with the urine, it can expand superiorly. It can expand so much that that it can go into the abdominal cavity also. When the bladder is empty, it lies in the pelvic cavity. When it's full with the urine, it can go into the abdominal cavity, right? Because it can distend itself. Okay, it can extend itself. Okay, it can hold its capacities. It can hold 1,000 ml of urine, right? When it's extended. Okay, and this makes it very ideal. And this is because it has a special type of epithelia called as the transitional epithelia. Okay, and when there is less urine here, so it will collapse. The way we spoke about, a stomach, when there is no food, it is thrown into ragi. Remember ragi in the stomach, right? So it's the same thing. When it collapses, when there is no urine, its wall collapses, it's thrown into ragi. And when there is a lot of urine, it will again flatten and the ragi will disappear. Okay. So, so what are the walls of the bladder made up of? Walls of the bladder are made up of three different types of layers. One is called as a mucosa, which is simply a transitional epithelium, which I just explained it to you. Okay. And then second is the smooth muscle. Okay. And those smooth muscles are called as the detrusor muscles. So these are special muscles present only in the bladder, where they are made up of three different types of the muscles: inner longitudinal layer, middle circular layer, and the outer longitudinal. So if you see, this is the opposite to what we discussed in the stomach. So you will have, sorry, it has to be the inner longitudinal row, right? And then you have the middle circular layer, okay, and then you have again the outer longitudinal layer. Okay, and these three are together called as the detrusor muscles. Okay, clear? Okay, a moderately full bladder is about 5 inches long and holds about 500 ml of the urine, but it has the capacity to hold about 1000 ml of urine. Okay, yes, that is the capacity of our bladder. It can hold about 1000 ml, but usually it's filled up to 500 ml. Okay, and it goes up, and when it's fully filled, it can go up into the abdominal cavity there. Okay, so then we have the urethra. Urethra is basically, as you can see here, this is the urethra for the females. Okay, it's a thin walled tube. Its main function is to carry urine from the bladder to the outside of the body. Okay, that is its main function. Okay, and again the process called as the peristalsis is very active in the in making sure that the urine goes out from the bladder into the outside the body. Okay, so what is the basic difference between the male urethra and the female urethra? There are two differences. One, female urethra is shorter, as you can see here, right? It's very shorter here. So that is why there's a chance of microorganisms. Females will usually suffer with urinary tract infections, UTI we call it, right? Because their urethra is a little shorter. So microorganisms can easily enter and occupy this trigone area. But look at the males. If you see here, this is the urethra for the males. The male urethra, look at this, how long it is, point number one. And male urethra has, through the urethra, both the urine as well as the sperms pass through the urethra. This is the basic difference between the male and the female urethra. See, female urethra is just 3 to 4 centimeters long, whereas a male is 20 centimeters long. So this is the basic difference between the two. Okay, and location also. Females have urethra anterior to the vaginal opening. Okay, while as a males travel, it travels through the prostate. I will tell you what is a prostate gland in a while. Okay, it travels through the prostate gland. Okay, and what about the epithelial tissue here? There is epithelia will change over here. We don't have transitional epithelia here. We have again the stratified squamous epithelia here. Okay, that is another difference, that the change of the, so here we had a transitional epithelia, but then in the urethra we have what? We have the what do you call it? We have the stratified squamous epithelia. Okay, and then what is a prostate? Okay, I don't know whether you have heard about it or not. So prostate is like, so this diagram is not in your book, but yes, prostate is there. Okay, so that's why I got this diagram. So there's a gland. Look at this one and this one. Okay, there's a pair of gland present in the males. Okay, which surrounds the urethra. As you can see, this is the urethra over here, right? So it surrounds the urethra. It is made up of almost 20 to 30 glands together and made up of dense connective tissue. Okay, and prostate produces base. It has nothing to do with the urinary system because it produces secretions for the sperms, right? So for the the volume of the semen, okay, is usually is basically decided by the prostate secretions. Okay, usually almost 1 by 3 of the volume of the semen, okay, basically comes from the prostate gland. So it's a gland basically in the males. Okay, did you understand what is the prostate gland now? And also one more thing, we have the two types of sphincters, the internal urethral sphincter, okay, and then we have the external urethral sphincter, right? This is one sphincter over here. Okay, and then we have one more sphincter over here. So this one is made up of smooth muscle and this one is made up of skeletal muscle. Okay, For the micturition, what is micturition? Micturition is when you want to go to washroom, when you want to empty the bladder, when we feel like going out to the bladder, right? So in order to urinate, right, what is important? Three things should happen. Okay. Number one, our detrusor muscle of the bladder. Okay, the detrusor muscle of the bladder should contract. Okay. So number one, because it's innervated with the sympathetic and the parasympathetic nervous system, right? So it can contract and send the urine out. Okay. And the second thing is that the opening of the internal sphincter, opening of this sphincter. So when this sphincter will open, then only we might be able to urinate. Okay. And then the last one is the opening of this sphincter, the external sphincter, which is a skeletal muscle. Okay. And this is controlled by the somatic nervous system. So based upon our will, if we really want to go to washroom, then only we can contract this muscle and urinate. And the process is called as the micturition. Okay. Urinating. Okay. And then we also have a lot of stretch receptors, okay, present everywhere. We have stretch receptors everywhere in the bladder, which take the information to the sacral region in the spinal cord. In return, the impulses travel back to the bladder through the pelvic splenic nerves, okay, which cause bladder contractions, and detrusor muscles can contract, and it can help in voiding the urinary bladder, or you can say micturition, emptying the bladder. Okay, this again the micturition. Okay, and then we stop over here. Okay, in tomorrow's class we will discuss the we just need to discuss about the hormones. We have almost spent covered majority of the chapter, but as I said that this chapter is very short, so let's see how much we can finish tomorrow, and accordingly I'll communicate it to you, right? I hope you're studying every day, because final exams are approaching. Okay, so do you have any question? Anybody has any question if you did not understand anything? Usually this chapter, students usually find this chapter. Are you Lian? I will take the attendance from the Zoom, Lian, don't worry. Yes, I'll take the attendance from the Zoom. Okay, so this chapter is usually students find it interesting, and it's a very short chapter. Okay, no issues. You can send me an email, Lian. I'll take care of it. Okay, so no questions? Okay, then everyone have a wonderful day ahead. Bye bye, everyone. Thank you, everyone. Bye bye.